All right, thank you all so much. I feel really lucky to close out the day for you all. It's been really inspiring to hear a lot of very cool things, and thank you to the organizers for putting this together. I know it's the first, but I think it's not going to be the last. So I wanted to share a little bit with you all about how the students got here. So these are the students in my upper level cellular and molecular neuroscience course. During their poster presentation, here's Loon the dog coming to provide external validation. So this is an external validation cure. There are also other faculty, and the students are invited to invite their friends, people that they love. We bring little snacks. So it's like a little party celebrating what they did. So when we design things, we always think about who we're designing it for, and these are who my students are. So I teach at a small liberal arts college. We have about 3,000 students. They're all undergraduates. And neuroscience is part of one of the big majors on campus, meaning we have 60 graduates each year. And in terms of our sort of racial, ethnic, and also gender demographics, we really have quite an ethnically and racially diverse group. So in 2024, we have about 40% of our majors are domestic students of color, and many are also international students. This course is an intermediate required course for the neuroscience major, or it can also be an upper level elective for biology majors. So most of the students in this class, by the time they get to their first neuro course and lab, have taken genetics to a sequence in chemistry. They've also taken biodiversity and evolution. So they've done a bunch of labs. They've done a bunch of different techniques. They've come to me knowing how to pipette. But this is the first time that they get to do a course in neuroscience. So now we don't just talk about cells, we're talking about neurons and glia. And they really, really love that. I think I feel really lucky that every year there's only 16 students in the course. And it is a 1 hour, they're 1 hour lectures Monday, Wednesday, Friday. And then what I'm telling you about today is how I use 3 hours every Tuesday over the course of about 15 weeks to do this lab. I get 2 undergraduate TAs who are really amazing, and we are also really fortunate to have staff who get to support, who provide a lot of support for prepping this lab. I'm really influenced by and inspired by a lot of disability justice advocates, and I think that, you know, in like higher ed, in academia, in sort of research science, we're only beginning to know how to adopt some of these disability justice frameworks. But I think our current sort of best estimate, the language that we do have right now, is thinking about universal design of learning, in which the goal here is an emphasis on learner agency. And that's done through creating these sort of experiences, right? So I'm not thinking at the level of assignments necessarily or lectures or things like this, but rather like this 15 weeks we have together, how am I able to create experiences that are really facilitating agency? I'm not going to go into details, but these are linked here if it's helpful. Okay, so here's what I did. I created a lab manual, which is also a little bit of a structure, I think, for some of the logics of how I wanted to help or reach this population of students, their first time in a neuroscience course doing a neuroscience lab. You are welcome to access it if you'd like and follow along. I'll leave this here for a little bit. And when you go to this link, it should take you to a Notion page, which is a really cool app that lets me design this very flexible wiki. So students can use it on their phone, tablets, their computers, whatever it is that they need. And as I was thinking about this lab, I wanted it to have some of the features of a really intensive research experience, but it's once a week, Tuesdays, 3 hours for about 15 weeks. But here are some of the features that I really wanted to adapt. One is I really wanted them to have this longer multi-step research experience. They've already done genetics lab, they've already done a bunch of chemistry labs where I think they sort of had these like disparate multiple labs whose purpose was to reinforce some of the content in lecture. But I wanted them to have this sort of longer cohesive research experience. I also wanted to make sure that they were exposed to techniques and tools that were used in contemporary neuroscience, and that they could answer a self-directed question. I wanted each student or student team to do every part of their own project. So this was really important to me because I think, sometimes, at least, speaking for my field, neuro can be very methods and technique based. And I just wanted to make sure that they knew that there was also, much of what we do in research is kind of like thinking, right? And planning, doing the logics of things. And I wanted them to make sure that when they do every part of the project, I think they get a chance to really see all of their sort of like, they get to test their own logics, right? And that was really, really important to me. I wanted them to have some practice in the dissemination of knowledge. And I also wanted them to learn how to work together in science around the science itself. So I didn't want them to like be friends necessarily in this sort of social icebreaker activity. Are they necessarily like friendly and with each other? I think that we really have this really beautiful opportunity that the science itself is actually facilitating social connections, right? So a couple of these things take time. This is like, this is sort of stuff that happens in our labs over the summer, right? We're 40 hours a week, they're with us. But I thought that, like, especially these parts, one of the things that I think many cures do a really good job of is a whole class might be together collecting different parts of data for the same project. And that's a little bit different than what I wanted to do here. So I was trying to figure out what were some ways that I can solve this problem. I decided that we were going to do a semester-long cure where we got to ask a research question at a cellular molecular level. I used a structured hypothesis development part to help them come up with a self-directed question. You can look at the lab manual for what that looks like. I wanted to give them I think they were sort of like traumatized from having to write a lab report in the earlier labs, and they hadn't had a chance to try this poster session. So we did that little poster session. And I also wanted them to work on these project teams. There were 4 students per group, and there was a lot of in-class time for structured and scaffolded teamwork planning and checking in. I won't talk about this quite a lot here. I'm happy to if there's more time. This is a really, really critical piece of the lab itself. And using existing open educational resources to do, the existing activities that are already out there really allowed me to help the students learn the tools and techniques we would be using in class. And that's what I'll be focusing on today. So there were 2 main existing OERs that I relied on. One is thanks to the Allen Education Materials Library. There is a really amazing exploring pathways in the brain activity, which helped me to help the students get introduced to using the Allen Brain Atlas. What we did in this lab is the students got to section mouse brains, and they basically used immunohistochemistry in order to identify protein of interest, as well as perineuronal nets, which they're not proteins, they're chondroitin sulfate proteoglycan, so they're like sugar protein structures, which also for people who do immunohistochemistry is a really great way to introduce students to like getting away from antibody language, right, by contrasting all the different ways that they can use that. Anyway, so this tool was really great because it let the students walk through the brain atlas. And then at the end of that, I could ask them, you have a hypothesis now about a brain region. Find the region that you're looking for. Take a photo of it or a screenshot of it so that you have that as a reference when you go to section. And they then have a lot more comfort with this Allen Brain Atlas, or brain atlases in general. The other thing that this tool did was it also helped them to explore the gene expression atlas. And so there are protein domains or different proteins that they would be interested in. And it allowed them to do this thing that we all do as scientists when we're interested in a hypothesis. We're like, has anyone looked at this yet? Like, is there even this protein in this brain region? And so that's really what they did. So the Allen tool helped them figure out how to use this. And then I asked them, check out one of the proteins associated with perineuronal nets. Is the region that you're hypothesizing has to do with whatever behavioral manipulation we did? Does it show that that protein is there? Take a screenshot of it, have it ready. So the other big tool that I think is really hard for students to learn is Fiji or ImageJ, a software that you might use to analyze like your histology slides. But there is, very fortunately, a really great tool that's already been developed by Bill Grissom. And what Bill has done is allowed us to use his lab's birdsong histology slides. And in this sort of little activity, they have given different doses of estrogen to male and female zebra finches. And they've given us access to those histology slides. And it's a structured activity that walks the students through using Fiji, downloading Fiji, but actually, I think it's still using ImageJ language, but you download it, how to use it. There are these tutorials, how to use it to measure all of the structures of interest. And I use this as a way of, so while they're waiting for their second incubation on the little shaker, they walk through this activity together with their group. And at the end of it, they learn how to use ImageJ or Fiji pretty well. And then I get to ask them, when you pull up one of your data, your like predicted data sets or your predicted images from the microscope, and try it out. Play around with it, and they feel a lot more comfortable. So just showing you some of the ways that using different kinds of OER activities in this more modular way as part of a broader thing that you might be trying to do in your classes can be a really both like time-saving way in order to teach students how to use the tools and techniques that are really critical so that we can focus on a lot of other things, right? Like I have time devoted in class where the students make a teamwork plan and then they check in. And in a typical cure where I feel like I have to teach them how to use Fiji, this is maybe the first thing to go, right? It can't be data collection because we want them to have that authentic experience. But I think using this, all of these OERs really allowed me to be able to free up time for much more other things. Okay. A couple of considerations when using OERs. It's great to be mindful of and respectful of how people want their resources to be used. This was something that was sort of newish to me when I first started developing OERs, is that many folks have already referenced Creative Commons licenses. So there are lots of different types of licensing, and it's just important to be very, very mindful. It's always also just good practice to attribute. Something that was brought up this morning is the doers. They've created this really amazing resource. I've just screenshot like one part of it, where they're really helping folks who do OER work think about how to talk about their work in reviews, promotion, and tenure. So this is, my brain thinks this way. I think this is amazing. It's the contribution. Let's say you use an OER in your class or classes. Here's some evidence that you can collect. And then this, they're saying, is sort of part of your teaching, part of your package, right? Or service, revising other OERs to be more relevant to student needs. There's the link here if you want to look at the full document. Okay. Thank you so much. I'm happy to answer any questions. I think we have plenty of time. And I wanted to just sort of give you all a little tidbit of some of my thinking around how to develop these things, some of the frameworks that I'm sort of bringing in. And you can look at any of the resources here on my website. It'll link to this slide, which will give access to also this Just a Start OER database, which includes cubes. Oh, yes, and the Allen Educational Materials Library.
All right, we do indeed have time for questions. While our mic runners grab their mics, I did just want to thank you for calling out that this can be a time-saving exercise in your classes. Building in these kinds of opportunities, the authentic opportunities, doesn't have to take more time than you were already spending.
Totally. Yep.
All right, where we got somebody? Great.
Thank you. This was really great. And I love how you share your protocols and resources. And this is a creative use of Notion.
Oh, yeah.
And a nice way of integrating media and videos and creating a presentation or a poster. One question I had, and I think Sam is going to laugh at me because he knows I was going to ask this eventually. What about lab notebooks and helping students document the process? Is there a way you could integrate that into your Notion setup or something else in a way that's open?
Yeah, that's such a good question. In the group work agreement, thank you so much for flipping to this. I can show you all. In the group work agreement, the students have to talk about what tools they want to use. And I usually let them sort of decide for themselves what their lab notebook format will look like. So they might share a Google Doc or like that kind of thing. Yeah. But I will say I don't give them much instruction about how to do a lab notebook. Again, I think I'm really lucky. This is a 300 level course, so that's all taught a little bit earlier in genetics. Mm-hmm.
Yeah.
Super impressive setup. I'm really astounded by this. I was just wondering how long it took you to put together all of those materials, and then I was also wondering if you have tested this multiple times or if you've just done this once, and if it ran completely smoothly or if you've adapted it a little bit.
Oh, sure. It definitely did not run completely smoothly. Yeah, so I have taught a version of this lab, I would say maybe 2021 was the first time. And then I set this up in Notion in 2022. And I've taught it once a year since. And yeah, I think I've included lots of research, student questions in the course evaluations at the end that are very specific, what did you like about this very specific thing? We also have a really cool mechanism at Macalester called mid-course interviews, where someone will come and do basically a focus group with your students. So I make sure they ask very specific questions about that. And then, I just adjust. But the first time I ran it, it really was kind of a nightmare. Like, I was like, I'm doing too much, which I think is actually what brought all of the things together. Yeah, and the students really like this Notion format. I mean, I was using it to sort of collect for my lab. My lab has a protocols wiki, which is also open if y'all want to look at it. But this was like for my lab. And then I was like, wait, why don't I just use this for this? And it turns out they really like it, that it frequently comes up on student course evaluations. It's also very flexible, so they can look at it for everything. It's pretty.
Last question.
So Notion is, tell me if I'm wrong, it's more like a WordPress site that allows you to create this site, right? How flexible this is? How much time does it take for you to kind of update your protocols and do all of the work on Notion? And have you compared it with any other site building platform?
It is, so I think that Notion is quite flexible. If you look on it, you can actually it has multiple uses. It's really meant for people to like track projects, but you can have meeting notes, goal trackers. I think it's trying to kind of be everything, like your one-stop shop for all the things. What I did was just choose Wiki, and I made it that way. I found it really easy. It's preferable to WordPress or other things because it's just a lot more flexible. Like I can show you all, if I wanted to add something, I can just add it. And you can choose a new template or, it's just very, like here, I can add all of these things. So it's extremely flexible. It did not take, it took a really long time to set it up, but most of the editing works really well. And I enjoy that it's very flexible. And it's also something I can share easily, openly. Yeah, I would recommend it. I am not sponsored by Notion, but.
Well, thank you.
Yeah, thank you all so much.
And